Apparently, signals from other cells in such niches keep the stem cell quiescent and undifferentiated. Stem cell niches have been identified in many organs. On the picture you see an adult stem cell that exists within niches where a rich interaction occurs between the stem cells and other niche cells, such as endothelial cells and adipocytes. In the brain, neural stem cells occur in the subventricular zone and dentate gyrus. In the skin, tissue stem cells are found in the bulge region of the hair follicle, and in the cornea, they are found at the limbus. Perhaps the most extensively studied tissue stem cells are hematopoietic stem cells found in the bone marrow. Although rare, they can be purified to virtual purity based on cell surface markers. Hematopoietic stem cells can be isolated from bone marrow, as well as from the peripheral blood after mobilization by administration of certain cytokines such as granulocyte colony stimulating factor. As is well known, they can give rise to all blood cell lineages and continuously replenish the former elements of the blood as these are consumed in the periphery. In clinical practice, marrow stem cells are used for treatment of diseases such as leukemia and lymphomas. In addition to hematopoietic stem cells, the bone marrow also contains a somewhat distinctive population of tissue stem cells, often called mesenchymal stem cells, and you see them on the picture. These cells can give rise to a variety of mesenchymal cells such as chondroblasts, osteoblasts, myoblasts, and others. Hence, there is great interest in their therapeutic potential. The ability to identify and isolate stem cells has given rise to the new field of regenerative medicine, which has as its main goal the repopulation of damaged organs by using differentiated progeny of embryonic stem cells or adult stem cells. Since embryonic stem cells have extensive self-renewal capacity and can give rise to all cell lineages, they often are considered ideal for developing specialized cells for therapeutic purposes. However, since embryonic stem cells are derived from blastocytes, typically produced from in vitro fertilization, their progeny carry histocompatibility molecules, human leukocyte antigen in people, of the donors of the egg and sperm. Therefore, they are likely to evoke immunologically mediated rejection by the host, just as organs transplanted from genetically disparate hosts do. Hence, much effort has gone into producing cells with the potential of embryonic stem cells from patient tissues. These cells are called induced pluripotent stem cells. To accomplish this goal, the expressed genes in embryonic stem cells and differentiated cells have been compared, and a handful of genes that are critical for the stem cellness of embryonic stem cells have been identified. Introduction of such genes into fully differentiated cells, such as fibroblast or skin epithelial cells, leads quite remarkably to reprogramming of the somatic cell nucleus, such that the cells acquire many of the properties of embryonic stem cells. Since induced pluripotent stem cells can be derived from each patient, their differentiated progeny should engraft successfully and restore or replace damaged or deficient cells in the patient. For example, insulin-secreting B cells in a patient with diabetes shown on the picture. Although these cells hold considerable promise, their clinical usefulness remains to be proved. In conclusion, we can say that regeneration of tissues is driven by proliferation of uninjured residual cells and replacement from stem cells. Cell proliferation occurs when quiescent cells enter the cell cycle. The cell cycle is tightly regulated by stimulators and inhibitors and contains intrinsic checkpoint controls to prevent replication of abnormal cells. Tissues are divided into labile, stable, and permanent according to the proliferative capacity of these cells. Continuously dividing tissues contain mature cells that are capable of dividing and stem cells that differentiate to replenish lost cells. Stem cells from embryos are pluripotent. Adult tissues, particularly the bone marrow, contain adult stem cells capable of generating multiple cell lineages. Induced pluripotent stem cells are derived by introducing into mature cell genes that are characteristic of embryogenic cells. The induced pluripotent stem cells acquire many characteristics of stem cells. Most growth factors are proteins that stimulate the survival and proliferation of particular cells and may also promote migration, differentiation, and other cellular responses. They induce cell proliferation by binding to specific receptors and affecting the expression of genes, whose products typically have several functions. They promote entry of cells into the cell cycle. They relieve blocks of cell cycle progression, therefore promoting replication. They prevent apoptosis, and they enhance the synthesis of cellular proteins in preparation of mitosis. A major activity of growth factors is to stimulate the function of growth control genes, many of which are called proto-oncogenes because mutations in them lead to unrestrained cell proliferation characteristics of cancer, also known as oncogenesis. There is a huge and ever-increasing list of known growth factors. In the following slides, rather than attempting an exhaustive cataloging, we highlight only selected molecules that contribute to tissue repair. And you see some of them on this table presented on the slide. Many of the growth factors that are involved in repair are produced by macrophages and lymphocytes that are recruited to the site of injury or activated at the site as part of the inflammatory process. Other growth factors are produced by parenchymal cells or stromal cells in response to cell injury. We start the discussion by describing general principles of growth factor actions. Most growth factors function by binding to specific cell surface receptors and triggering biochemical signals in cells. The major intracellular signaling pathways induced by growth factor receptors are similar to those of many other cellular receptors that recognize extracellular ligands. In general, these signals lead to the stimulation or repression of gene expression. Signaling may occur directly in the same cell that produced the factor and is known as autocrine signaling between adjacent cell, and this is an example of paracrine signaling shown in the middle of the picture, or over great distances, and this is an example of endocrine signaling shown on the right. Receptor proteins are generally located on the cell surface, but they may be intracellular. The ligands must be sufficiently hydrophobic to enter the cell. On the basis of their major signaling transduction pathways, plasma membrane receptors fall into three main types: receptors with intrinsic kinase activity, G-protein coupled receptors, receptors without intrinsic enzymatic activity. Binding of ligand to the extracellular portion of the receptor causes dimerization and subsequent phosphorylation of the receptor subunits. Once phosphorylated, the receptors can bind and activate other intracellular proteins like phosphate idylanisitol kinase, phospholipase, And they also stimulate downstream signals that lead to cell proliferation or induction of various transcriptional programs. The G protein-coupled receptors contain 7 transmembrane alpha helix segments and are also known as 7 transmembrane receptors. After ligand binding, the receptors associate with intracellular guanosine triphosphate binding proteins that contain guanosine diphosphate, GDP. Binding of the G proteins causes the exchange of guanosine diphosphate with guanosine triphosphate, resulting in activation of the proteins. Among the several signaling pathways activated through the G protein-coupled receptors are those involving cyclic adenosine monophosphate or 3 and 5 cyclic adenosine monophosphate (CAMP), and the generation of inositol 1,4,5-triphosphate, which releases calcium from endoplasmic reticulum. Receptors in this category constitute the largest family of plasma membrane receptors. There are more than 1,500 members that have been identified so far. The receptors without intrinsic enzymatic activity are usually monomeric transmembrane molecules with an extracellular ligand-binding domain. Ligand interaction induces an intracellular conformational change that allows association with intracellular protein kinases called Janus kinases. Phosphorylation of Janus kinases activates cytoplasmic transcription factors called STATs, signal transducers and activators of transcription, that shuttle into the nucleus and induce transcription of target genes. In conclusion, we can say that polypeptide growth factors act in autocrine, paracrine, and endocrine manner. Growth factors are produced transiently in response to an external stimulus and act by binding to cellular receptors. Different classes of growth factor receptors include receptors with intrinsic kinase activity, G protein-coupled receptors, and receptors without intrinsic kinase activity. Growth factors such as epidermal growth factor and hepatocyte growth factor bind to receptors with intrinsic kinase activity, triggering a cascade of phosphorylating events from MAP kinases, which culminate in transcription factor activation and DNA replication. G protein-coupled receptors produce multiple effects via the CAMP and calcium pathways. Chemokines utilize such receptors. Cytokines generally bind to receptors without kinase activity. Such receptors interact with cytoplasmic transcription factors that move into the nucleus. Most growth factors have multiple effects, such as cell migration, differentiation, stimulation of angiogenesis and fibrogenesis, in addition to cell proliferation. Tissue repair depends not only on growth factors activity but also on interactions between cells and extracellular matrix components. The extracellular matrix is a complex of several proteins that assembles into a network that surrounds cells and constitutes a significant proportion of any tissue. Extracellular matrix sequesters water, providing turgor to soft tissues, and minerals, giving rigidity to bone. It also regulates the proliferation, movement, and differentiation of the cells living within it by supplying a substrate for cell adhesion and migration and serving as a reservoir for growth factors. The extracellular matrix is constantly being remodeled. Its synthesis and degradation accompany morphogenesis, wound healing, chronic fibrosis, and tumor invasion and metastasis. Extracellular matrix occurs in 2 basic forms: interstitial matrix, shown by red arrows, and basement membrane, shown by the black arrow. Interstitial matrix is present in the spaces between cells in connective tissue and between epithelium and supportive vascular and smooth muscle structures. It is synthesized by mesenchymal cells and tends to form a three-dimensional amorphous gel. Its major constituents are fibrillar and non-fibrillar collagens, as well as fibronectin, elastin, proteoglycans, hyaluronate, and other elements. The basement membrane. The seemingly random array of interstitial matrix in connective tissues becomes highly organized around epithelial cells, shown by the red arrow, endothelial cells, and smooth muscle cells, forming the specialized basement membrane. The basement membrane lies beneath the epithelium and is synthesized by overlying epithelium and underlying mesenchymal cells. It tends to form a plate-like chicken wire quote-unquote mesh. Its major constituents are amorphous non-fibrillar type 4 collagen and laminin. There are three basic components of extracellular matrix: fibrostructural proteins such as collagen and elastins, which confer tensile strength and recoil; water-hydrated gels such as proteoglycans and hyaluronan, which permit resilience and lubrication; and adhesive glycoproteins that connect the matrix elements to one another and to cells. The collagens are composed of three separate polypeptide chains braided into a rope-like triple helix. Approximately 30 collagen types have been identified, some of which are unique to specific cells and tissues. Some collagen types form fibrils by virtue of lateral cross-linking of the triple helices. The fibrillar collagens form a major proportion of the connective tissue in healing wounds and particularly in scars. The tensile strength of the fibrillar collagens derive from their cross-linking, which is the result of covalent bonds catalyzed by the enzyme lysyl oxidase. This process is dependent on vitamin C. Therefore, individuals with vitamin C deficiency have skeletal deformities, bleed easily because of weak vascular wall basement membrane, and suffer from poor wound healing. Genetic defects in these collagens cause diseases such as osteogenesis imperfecta and Ehlers-Danlos loss syndrome. Other collagens are non-fibrillar and may form basement membrane type 4 or be components of other structures such as intravertebral discs type 9 or dermal epidermal junctions type 7. The ability of tissues to recoil and return to a baseline structure after physical stress is conferred by elastic tissue. This is especially important in the walls of large vessels, which must accommodate recurrent pulsatile flow, as well as in the uterus, skin, and ligaments. Morphologically, elastic fibers consist of a central core of elastin, surrounded by a mesh-like network of fibrillin glycoprotein. Defects in fibrillin synthesis lead to skeletal abnormalities and weakened aortic walls, as in Marfan syndrome. Proteoglycans form highly hydrated compressible gels, conferring resilience and lubrication. They consist of long polysaccharides called glycosaminoglycans or mucopolysaccharides, including dermatan sulfate or heparan sulfate, linked to a protein backbone. Hyaluronan, also called as hyaluronic acid, a huge mucopolysaccharide without a protein core, is also an important constituent of extracellular matrix that binds water and forms a viscous, gelatin-like matrix. Besides providing compressibility to tissues, proteoglycans also serve as reservoirs for growth factors secreted into the extracellular matrix. Some proteoglycans are integral cell membrane proteins that have roles in cell proliferation, migration, and adhesion, for example, by binding growth factors and chemokines and providing high local concentrations in these mediators. Adhesive glycoproteins and adhesion receptors are structurally diverse molecules involved in cell-to-cell adhesion, shown on the picture, the linkage of cells to the extracellular matrix, and binding between extracellular matrix components. The adhesive glycoproteins include fibronectin, a major component of the interstitial extracellular matrix, and laminin, a major constituent of basement membrane. The adhesion receptors, also known as cell adhesion molecules, CAMs, are grouped into 4 families: immunoglobulins, cadherins, selectins, and integrins. Fibronectin is a large disulfide-linked heterodimer synthesized by a variety of cells, including fibroblasts, monocytes, and endothelium, that exists in tissue and plasma forms. Fibronectins have specific domains that bind to a wide spectrum of extracellular matrix components, including collagen, fibrin, heparin, proteoglycans, and can also attach to cell integrins via a tripeptide arginine glycine aspartic acid motif, abbreviated as RGD. Tissue fibronectin forms fibrillar aggregates at wound healing sites. Plasma fibronectin binds to fibrin within the blood clot that forms the inner wound, providing the substratum for extracellular matrix deposition and reepithelialization. On the picture you see the fibronectin molecule consists of repeating globular domains with specific cell binding sites for interactions with integrins. 4 of fibronectin peptide sites that can interact with specific integrins are shown extending down toward the integrins. Integrins contain 1 alpha and 1 beta subunit, which span the plasma membrane and terminate in short cytoplasmic domains that are thought to mediate intracellular signaling. Laminin is the most abundant glycoprotein in basement membrane. It is a cross-shaped heterotrimer that connects cells to underlying extracellular matrix components such as type 4 collagen and heparan sulfate. Besides mediating attachment to basement membrane, laminin can also modulate cell proliferation, differentiation, and motility. Integrins are a family of transmembrane heterodimeric glycoprotein chains that were introduced in the context of leukocyte adhesion to endothelium. They are also the main cellular receptors for extracellular matrix components, such as fibronectins and laminins. We have already talked about some of the integrins as leukocyte surface molecules that mediate firm adhesion and transmigration across endothelium at sites of inflammation, and we shall meet them again when we discuss platelet aggregation. Integrins are present in the plasma membrane of most cells, with the exception of red blood cells. They bind to many extracellular matrix components through arginine glycine aspartic acid motifs, initiating signaling cascades that can affect cell locomotion, proliferation, and differentiation. The intracellular domains link to actin filaments, thereby affecting cell shape and mobility. The extracellular matrix is much more than a space filler around cells. Its various functions include mechanical support, control of cell proliferation, scaffolding for tissue renewal, establishment of tissue microenvironments. Mechanical support for cell anchorage and cell migration and maintenance of cell polarity. Control of cell proliferation by binding and displaying growth factors and by signaling through cellular receptors of the integrin family. The type of extracellular matrix proteins can affect the degree of differentiation of the cells in the tissue, again acting largely through cell surface integrins. On the picture you see formed unpatterned tissue with many folds on the cell surface due to a spontaneous lethal mutation and completely loss of signaling molecules of differentiation. Because maintenance of normal tissue structure requires a basement membrane or stromal scaffold, the integrity of the basement membrane of the stroma of the parenchymal cells is critical for the organized regeneration of tissues. Therefore, although labile and stable cells are capable of regeneration, disruption of the extracellular matrix results in a failure of the tissues to regenerate and repair by scar formation. On the picture you see remodeling of the lung extracellular matrix. During progression of chronic obstructive pulmonary diseases, the basement membrane and the interstitial matrix is remodeled, resulting in a disruption of the interface between these two extracellular matrix compartments. Type 6 collagen is situated in this interface, and consequently fragments of type 6 collagen may be particularly related to disease progression and lung tissue remodeling. In addition, the chronic inflammatory response may in part be accompanied by persistent neutrophil presence in affected tissues, which normally would be resolved in the later stages of inflammation resolution. These inflammatory cells produce high amount of neutrophil elastase capable of degrading the elastin of the lungs found in the interstitial matrix, which is essential for lung tissue elasticity. Both the type 6 collagen and the elastin remodeling, in addition to the remodeling of other ECM components, results in the release of small protein fragments to the bloodstream. Basement membrane acts as a boundary between epithelium and underlying connective tissue, and also forms part of the filtration apparatus in the kidney. On the right picture, you see the glomerular filtration system, glomerular tuft, the walls of which constitute the filter system. On the top and on the bottom is the glomerulus. The plasma filtrate is led to the proximal tubule, and the unfiltered blood returns to the blood circulation. The filtration barrier of the capillary wall contains the innermost fenestrated endothelium, the glomerular basement membrane, and the podocytes with their interdigitating food processes. The slit diaphragm is a uniformly wide porous filter structure located between the food processes. To date, the slit diaphragm has been shown to contain distinct components such as nephrin, p-cadherin, NEF1 and fat, but how these proteins build up the structure is still unknown. In conclusion, we can say that extracellular matrix consists of the interstitial matrix between cells made up of collagens and several glycoproteins, and basement membranes, underlying epithelia and surrounding vessels, made up of non-fibrillar collagen and laminin. The extracellular matrix serves several important functions. It provides mechanical support to tissues. This is the role of collagens and elastin. It acts as a substrate for cell growth and the formation of tissue microenvironments. It regulates cell proliferation and differentiation. Proteoglycans bind growth factors and display them at high concentration, and fibronectin and laminin stimulate cells through cellular integrin receptors. An intact extracellular matrix is required for tissue regeneration, and if the extracellular matrix is damaged, repair can be accomplished only by scar formation. The importance of regeneration and replacement of injured tissue varies in different types of tissues and with the severity of injury. In liable tissues, such as the epithelia of the intestinal tract and skin, injured cells are rapidly replaced by proliferation of residual cells and differentiation of tissue stem cells, provided the underlying basement membrane is intact. The growth factors involved in these processes are not defined. On the left picture, you see a thin layer of epidermal reepithelialization shown by the red arrow and extensive granulation tissue formation in the dermis shown by the yellow arrow. On the right picture, you see continuing reepithelialization of the epidermis shown by the stem and wound contraction shown by the green arrow. On the picture on the left, you see also the skin, the blue arrow, commonly found in diabetic patients. On the right picture, you see a skin ulcer with a large gap between edges of the lesion. Loss of blood cells is corrected by proliferation of hematopoietic progenitors in the bone marrow and other tissues driven by colony-stimulating factors, which are produced in response to the reduced number of blood cells. On the picture, you see representation of the classical hierarchic model of hematopoiesis from the endosteal niche in the upper part of the figure to the blood vessel. Passing from the vascular niche, the following distinct population are depicted. The cells abbreviated as LT-HSC are long-term hematopoietic stem cells. ST-HSC, short-term hematopoietic stem cells. LSC, leukemic stem cells. MPP, multipotent progenitor. CLP, common lymphoid progenitor. CMP, common myeloid progenitor, and the cells between the committed progenitors and mature cells. Tissue regeneration can occur in parenchymal organs with stable cell population, but with the exception of the liver, this is usually a limited process. Pancreas, adrenal, thyroid, and lung have some regenerative capacity. On the picture, you see regeneration of the exocrine pancreas. The exocrine pancreas is composed of acinar cells that synthesize and secrete digestive enzymes, ductal cells that funnel the enzymes into the small intestine, and central acinar cells. The exocrine pancreas can regenerate spontaneously in humans by redifferentiation of acinar cells. Inflammatory injuries to the exocrine pancreas, such as acute pancreatitis, lead to acinar cell deaths. Loss of islet beta cells in diseases such as type 1 diabetes requires therapeutic intervention. The surgical removal of a kidney elicits in the contralateral kidney a compensatory response that consists of both hypertrophy and hyperplasia of proximal duct cells. The mechanism underlying this response is not understood well. The regenerative response of the liver that occurs after surgical removal of hepatic tissue is remarkable and unique among all organs. As much as 40 to 60% of the liver may be removed in a procedure called living donor transplantation, in which a portion of the liver is resected from a normal person and transplanted into a recipient with end-stage liver disease, or after partial hepatectomy performed for tumor removal. In both situations, the removal of tissue triggers a proliferative response of the remaining hepatocytes and the subsequent replication of hepatic non-parenchymal cells. On the pictures, you see regeneration of the liver. Computed tomography scans Show a donor liver in a living donor liver transplantation. On the left, the donor liver before the operation. Note the right lobe, which will be resected and used as a transplant. On the right is the scan of the same liver 1 week after resection of the right lobe. Note the enlargement of the left lobe with regrowth of the right lobe. In experimental systems, hepatocyte replication after partial hepatectomy is initiated by cytokines that prepare the cells for replication by stimulating the transition from G0 to G1 in the cell cycle. Progression through the cell cycle is dependent on the activity of growth factors such as hepatocyte growth factor produced by fibroblast endothelial cells and liver non-parenchymal cells, and the epidermal growth factor family of factors, which includes transforming growth factor alpha produced by many cell types. A point worthy of emphasis is that extensive regeneration or compensatory hyperplasia can occur only if the residual connective tissue framework is structurally intact, as after partial surgical resection. By contrast, if the entire tissue is damaged by infection or inflammation, regeneration is incomplete and is accompanied by scarring. For example, extensive destruction of the liver with the collapse of the reticulin framework, as occurs in a liver abscess that leads to scar formation, even though the remaining liver cells have the capacity to regenerate. We will stop here today and will continue during the next class. Thank you for your attention.